All right, we do. We've got several people here. Well, I would like to welcome everybody to Mohonk Preserve's webinar on restoring the American chestnut. My name is Lauren Bohr, and I am the Education Outreach Coordinator. I'm going to be monitoring, facilitating this whole presentation. And before I introduce our presenters, we have kind of a dual presenting here. We have Hannah Pilkey and we have Gail Whistance with us this evening. I just have a few reminders for everyone. I'm sure you've attended several Zoom things and webinar things, but please do turn off your video and mute your audio. So this is going to help prevent any distractions to our presenters and the others in the audience. If you do have questions, you can use the chat box. And I'll monitor those, and we can, there'll be a session at the end where we can answer lots of questions. And maybe you won't think of one until the end either, but there will be a chance to ask questions at the end. So as I mentioned, this presentation is going to be recorded along with the chat conversation to share with our online presence. So this will be available for you. I can send out a link. It will also be posted on our website and our social media as well. So we have, as I mentioned, we have a couple presenters this evening. Our main presenter is Hannah Pilkey, and we'll also have Gail Whistance with us. So Hannah has been working with the American chestnut tree for the past 5 years with SUNY-ESF's American Chestnut Research and Restoration Project in Syracuse. That's a long title. She's finishing up her master's degree and studying pollen collection and freezer storage to be used for pollinating. She's also the tree breeding and plant tissue culture production manager for the project and manages the clonal propagation and controlled outcrosses of the chestnut. In her spare time, she's the project's beekeeper, which is an awesome spare time activity. And Gail is originally from Illinois, but is a longtime Hudson Valley resident. She has been a member of Mohonk Preserve for over 40 years and the American Chestnut Foundation for almost 15 years. Her career was as a technical writer and programmer for IBM, but as a lot of us will eventually do, maybe in retirement, she's devoted much of her time to native landscaping and local history. So in 2015, she recruited a group of master gardener volunteers from the Cornell extension of Ulster County to plant a small orchard of American chestnuts on the Mohonk Preserve at Spring Farm. So she's going to tell us a little bit about that during her part of our presentation. So without further ado, I'm going to turn things over to Hannah and go ahead and enjoy.
Thank you so much. Well, first, I'd like to extend a thank you to you, Lauren, for hosting me today, and then also to Gail for getting me in contact with you guys, and also to the audience today. I mean, as bummed as I am not being able to meet with you guys in person, it's pretty cool that we can, you know, talk about cool naturey stuff and trees and interact from the comfort of our own living room. So I'm really excited to be here to share with you guys a little bit about our research project at ESF. Let me just share my screen real quick. Can everybody see? Is that looking good?
Yeah, I can see it.
Awesome. And then things are pretty casual, obviously. I mean, so if there's something that is like super confusing, sometimes I have a tendency to use jargon that can be unfamiliar, really confusing. So if you could just send something in the chat, I might not be able to see it, but feel free, like, Lauren, if you want to butt in and be like, hey, could you explain that real quick? Like, people are confused, like, please feel free.
Yes,
I'm going to minimize.
I'll monitor the chat for you.
Great, thank you so much. I'm going to minimize you guys to the side because it's kind of weird. I always get like distracted by myself talking, so I'm just going to hide this real quick before I get started. Okay, so today I'm going to be talking to you guys about the return of the American chestnut tree, and specifically ESF's part in it, which is using biotechnology as a tool for conservation. A little bit about our research project. As Lauren mentioned, we are the American Chestnut Research and Restoration Project, which is a mouthful to say. We are a nonprofit restoration program, which has been going since about 1990. And we are also partners and collaborators with the American Chestnut Foundation. So we were talking earlier, there's some members of the American Chestnut Foundation, the what I'm assuming is the New York chapter members. So they're also a nonprofit organization that has chapters in every state in the range of the American chestnut. It's a really incredible organization that we work with. But these are all the people that are in our research project in our lab. We're a pretty big research team. We're led by Dr. Bill Powell, he's our director, but this work is pretty much the work of many different graduates, undergrads, high schoolers, volunteers, and collaborators. So it's really the work of so many people over the past 30 years. And I know that there's already some chestnut enthusiasts in the crowd, but I have to, of course, give the obligatory background spiel about the American chestnut and its really kind of tragic background. So the American chestnut was once this really massive, like iconic tree that grew in the eastern part of our country. It was nicknamed the redwood of the East because it just grew to incredible heights. So some were recorded as being 100 feet tall. They had these giant diameters, 6 to 10 inches. You can see in these historical photos just how massive this tree was compared to the people standing next to it. So just a pretty iconic species. And then not only was it just a massive tree, it was also really dense and prolific. So in areas like in New York State, some of the composition of the tree would be like 1 in 25 or 1 in 4 American chestnuts. So about 25% of the forest was American chestnut. And then in some areas, like where the chestnut was really prolific, like in the Appalachian range, there were stands that were almost 100% American chestnut tree, because this guy was just a really prolific grower. And you can see in this picture here, actually, let me see if my pointer works. I hope it does. Ooh, it does. All of these white parts on the tops of the trees, this was all flowers of the American chestnut. So it was a really prolific flowerer, to the point where someone was quoted in a historical document saying that it was like snow in July when these trees would bloom. That's how dense they were. And because this tree was such a dense flowerer, it was also a really heavy nut crop producer. So this is kind of a unique feature to trees. Not all trees can produce nuts each year. So the chestnut not only produced nuts every year, but like a huge number of nuts. So these are more historical drawings of people going chestnutting and collecting chestnuts for all sorts of purposes. These are really cool. Actually, Gail sent us the middle one, which is a really lovely watercolor photo of an American chestnut and people gathering nuts off the ground. So this tree was really valuable to the people that lived among the tree. Of course, they collected the nuts for personal consumption, but they would also gather it to be sold in big cities. Apart from the nuts, they also used the timber for a bunch of different purposes. It was called the cradle to grave tree because you would use it for constructing your crib all the way to constructing your coffin, as was quoted in other historical documents. They used it for a lot of outdoor purposes because the tree was really rot resistant and durable. So you could use it for like your constructing fence posts and railroad ties, things of that nature, barns, stuff that was exposed to the elements because this tree is just so durable. And of course, there was a bunch of forest critters that really relied on this tree and had a really valuable relationship with it because the American chestnut was considered a keystone forest species. So that means it had a dramatic impact on the environment in which it grew, and it had the ability to alter the landscape where it grew pretty well, because it was really an important tree there. So of course, there were a bunch of different creatures that ate that edible mast crop every year, like bears and mammals. Even like turkeys could swallow chestnuts whole, which is pretty incredible that they could digest the shell of the chestnut. I think blue jays also were another bird species that loved chestnut. And then other creatures like insects relied on things like the flowers because they would eat the pollen and the nectar of the chestnut, and then the sap from the leaves and the bark. And the chestnut was also very important in other ways we don't think about directly, like when the leaves of the chestnut would fall off the tree, it would create, a really unique forest litter composition, and the leaves would fall into streams and create important habitat for stream critters like trouts and macroinvertebrates. And then even underground, all different plant species host mycorrhizal fungi. So the chestnut had unique fungi, and then of course that impacts the soil microinvertebrates. So it was really just an important tree for a dynamic forest species. And even though the chestnut's not around anymore, it's still pretty culturally significant. You can find little remnants of chestnut-related things all around you. You guys might have noticed during Christmas time, the famous Christmas song opening, chestnuts roasting on an open fire. Or if you go to pretty much any major city in the United States, there's a chestnut street. You can still find, because the wood is so durable, you can still find furniture and barns constructed of chestnut wood. And you can even find it like craft fairs, or if you do a quick search on Etsy, there's all sorts of materials that can be made from wormy reclaimed chestnut wood. When you see wormy chestnut, fun fact, that just means it was eaten partially by insects, because a lot of this, reclaimed wood was from, 100 years ago. So obviously there's been like a little insect damage, but I think it adds a little bit of character to chestnut wood products. And then of course, I always like to point out that there's a chestnut emoji on your smartphone. So this gets heavily used on our social media and in our emails and stuff. We just love this emoji, obviously. And part of the reason why the chestnut is still relevant today is because there's more than just the American chestnut that it comprised in the Castanea genus, which comprises all the chestnut species. So of course, we have our American chestnut here in North America, but we also have 3 other major chestnut species, and that includes the 2 Asiatic species, the Japanese and the Chinese chestnut. And then there's also the European chestnut tree too. And that's why you can still find chestnuts at the grocery store around the holidays, is because they're being imported from species that weren't decimated by this nasty fungus. And actually, it was our love for the chestnut which led to its demise, in that we loved the American chestnut so much that in the late 1800s and the early 1900s, people were like, oh, let's bring over the other species of chestnuts because they're also beautiful and produce nuts, and we can use them as horticulture crops. So they brought over Asiatic species to North America. And unfortunately, this brought the invasive fungal species Cryphonectria parasitica with it. Cryphonectria parasitica evolved naturally with the Chinese chestnut and the Japanese chestnut over millions and millions of years. So in the presence of this fungus, they kind of had this, defenses against each other. Like they would evolve, and the chestnut would figure out how to defend itself against this fungus. So when this fungus got to America, where the tree never lived with the fungus, of course, there was no defense in place to protect itself against this fungus. So the way that fungus works is that it invades wounds on the chestnut tree. So it can be something like a little bug chewing on the bark or a woodpecker. Even chestnuts are really fast growing species. So even like the little micro tears in the bark from when the chestnut grows is a mode of entry for this nasty fungus. So the fungus will make its way into the wounds, and it will secrete a really nasty substance called oxalic acid. You'll want to remember this because it'll come up later in the presentation. This oxalic acid, it works by lowering the pH of the bark of the chestnut, killing the tissue. And then the fungus is a necrotroph, so it comes in and it munches away at all the dead tissue, and it creates just these really awful, terrible cankers on it. And the cankers effectively girdle and then kill the chestnuts. So you can see here, the blight was discovered in 1904 in New York City, even though they thought it was probably a little bit earlier that the blight actually arrived. Also in this image, you can see just the natural range of the chestnut. So really just encompass the entire eastern part of the United States and into Canada, which isn't shown on this map. And then over the next 50 years, the blight made its way through this entire range of the chestnuts. And unfortunately, it had a severe impact on the 2 other North American Castanea species. So these are kind of more isolated, lesser known, I guess, chestnut species compared to the American chestnut, but they were also heavily hit by this blight fungus by the 1950s. And early on, there was a lot of effort to try to stop this fungus and its spread. There was quarantines, people cut down the infected trees, but unfortunately, the blight also survives on oaks and other tree species, so it can persist in the forest and just kind of hang out even when there's no chestnuts there. So it was once it was here, it's never going away. It's in our forest to stay. And then as the tree spread through the range of the chestnut, it created these really apocalyptic, terrible scenes, kind of kind of like how we see with the ash trees, except in some parts, especially in the Appalachian area, you know, you think about those 100% American chestnut stands where this is what was left of them. And of course, people held such a special relationship with these chestnuts that it was really devastating to them to see this. And they thought that there was no hope for the American chestnut tree, and it was a goner. And that is about as bummer as I'll be in this presentation. And now for the much more uplifting part of the presentation. I always get like, you know, kind of bummed talking about this, but on a positive note, the American chestnut tree is a really resilient species. It has an ability, it's called stump sprouting. So when the main part of the chestnut tree dies back, as you can see this part that was cut down here, it will release a bunch of new little sprouts from the bottom of the tree. So this is how the tree is still able to live now. The only problem is these sprouts will get big enough and then it'll just get reinfected by the blight. So it's kind of stuck in this really cyclical, awful death cycle that it can't escape. So you can still find chestnuts in pretty much anywhere in its natural range still, because they can do the stump sprouting. They just look like, we were talking about this earlier, they just look kind of like these shrimpy, sad little things, not the iconic, giant, massive redwoods of the East tree that it once was. So because of this, it has pretty much allowed 100 years of research on this tree to try to figure out ways to rescue it and bring it back to its former glory. The research project that we're on is just one facet of that, but there's been all sorts of research that has gone into protecting this tree. And one of which is the American Chestnut Foundation's back cross breeding program. So this is one avenue of conservation of the tree. So some of you guys might be familiar with this. I'll try to be brief in explaining it. This is essentially when you hybridize the American chestnut tree with the Chinese chestnut tree. Now, Chinese chestnut has that natural resistance because it evolved with the fungus over millions of years. So it has some sort of genetic component to it that's giving defense to the fungus. So what you do is you hybridize those two, and then you take the offspring hybrid, and then you back cross it to other American chestnut trees. So that means you breed it to a different American chestnut. And then each series of back crossing that you do, you're hopefully retaining the part of the genome of the Chinese chestnut tree that is giving the tree resistance while making it more American. So the goal of the TACF is to produce a tree that is 15/16 American chestnut, with only a 16th of it being Chinese. So you're ending up with a mostly American tree with a little bit of Chinese genes in there to hopefully give it resistance. But with any sort of research project, there's always, you know, things that make things difficult. And one of which is, with this program, is selective breeding, which is really difficult. When you're trying to breed for pathogen resistance, it can be really hard to select which trees and measure them and gauge which ones are resistant. And then originally, when this program was started, they thought that maybe 1 or 2 genes was giving the Chinese chestnut resistance to the fungus. But now with more genomic work, and as they're doing more research into them on a genomic level, they're figuring out that it could be up to 9 different genes that are giving this tree resistance. So the more genes that you have somewhere in that genome, the harder it is to capture that through selective breeding. And then, of course, a question that's always been postulated with this program is that are hybrids most ideal for forest restoration? So if you end up with a tree that's partially Chinese, Chinese chestnuts are, of course, you know, adapted to Chinese forests. So they're a little bit smaller and their nuts are a little bit bigger. So if you were to have some Chinese characteristics in there, along with hopefully blight resistance, you might be ending up with a slightly smaller American chestnut tree, or maybe one with a little bit bigger of nuts. So can these trees compete as well as a pure American chestnut tree, you know, that can grow as tall and still have the nuts that are small enough to be swallowed by turkeys, those sorts of characteristics. So because of these questions, the members of the New York chapter brought this question to my supervisor and his colleague at the time, which was, can genetic engineering be used to give the American chestnut tree resistance to the fungus? Because genetic engineering has been traditionally used to insert very precisely genes that you're interested in delivering to a plant species that one of the genes could be giving the tree resistance to the fungus. So this question was brought up, and then over the next 30 years, pretty much, Dr. Bill Powell and Dr. Chuck Maynard pretty much worked out which genes could possibly give chestnut trees, defense against the blights, and pretty much modes of getting the gene into the tree. It took many, many decades of figuring out how to do this. And I'll spare you guys the nitty-gritty details of all the methods for this, but we can talk about it at the end if anyone has questions on like how we actually insert the gene into the tree. But essentially, genetic engineering, they had selected a gene to put into the chestnut tree, and that is how the blight-tolerant darling American chestnut tree was created. And that's the tree that is currently going through federal regulatory review that is showing a lot of promise and being resistant to the fungus. So the gene that Bill and Chuck decided to go with after, reading research and, all the other research with different plants showed was that a gene called oxalate oxidase was a good candidate. Oxalate oxidase is a really ubiquitous enzyme. The gene codes for an enzyme, which is found in all sorts of different plants like bananas and strawberries and grasses, tons of different plants. The one that was cloned and put into the American chestnut genome was cloned from the wheat plant. And how it works is it detoxifies that really nasty oxalic acid that I mentioned earlier, which is produced by the fungus. So here's just like a little chemical breakdown of what it does. Here's the nasty fungus acid. And then here's the oxogene, which is an enzyme that's coming in and it's breaking down this nasty acid into hydrogen peroxide and carbon dioxide, which are both byproducts that are beneficial to a plant. And it's really important to note that the oxogene is not a pesticide. It's not killing the fungus in any way. It's allowing it to live there. It's just pretty much changing its lifestyle from a pathogen to a saprotroph, kind of like how it lives on a Chinese chestnut tree and some oak trees. It's just kind of putting it on a restricted diet so it can't go wild and just eat all of that tissue like it can with trees that don't have the oxogene. So after the darling tree was created, after they had all the kinks worked out and how to get the gene into the tree, they wanted to start testing it and putting it up against the fungus to see how it would respond. So here was one of the assays that was done with the fungus. So here over to the left, you can see these are the wild type American chestnut trees. So these are the trees that do not have the oxogene inserted into its genome. And pretty much all of them, 6 weeks after they were inoculated with the fungus, they had died. And you can see some of them actually starting to resprout from the bottom. And then in comparison to the darling American chestnut trees, which have the gene for blight resistance, the oxogene, all doing fabulous after 6 weeks of inoculation. And they actually, I think, were all still healthy about a year after this photo was taken. So just a really dramatic difference in response to the fungus. And then we also, in our experiments, use Chinese chestnut as a control because it has that natural resistance to the fungus. So this one, actually, 3 of them died in this photo, and 3 of them are still looking healthy. So it kind of had an intermediate response to being inoculated with the fungus. So Darling 58 was produced in tissue culture, which means we can clonally propagate it and produce many, many genetically identical trees in a tissue culture production lab. So we grow the trees in these, they're called magenta cubes, and they have all of this nutritious plant media that grows in the bottom of it. And then these plants can be propagated like crazy because you can use all these different node segments of the American chestnut. Anywhere there's a node can grow into a new tree in tissue culture. So we can take these and propagate them really easily. So this is what was done with Darling 58. They get planted out. So these are all clones that are growing in our growth chambers. So these are like little baby chestnuts. And then the majority of these cloned darling trees don't go out planted into the field. A lot of them were used for biosafety analyses, which I'll be touching on shortly, because you don't want to plant just a bunch of clones out into the field. That would be bad if a different pest came in and or something like climate change, where new adverse conditions were affecting these chestnuts. You wouldn't want clones being used for restoration. So a lot of these tissue culture propagated clones were put into highlight growth chambers to produce pollen, which is what you see here. If you give the chestnuts a lot of light and high intensity light, it will essentially stress them into producing pollen for you. So chestnuts typically take like 3 to 8 years to be sexually mature. Well, these guys that are under a high light treatment will produce pollen in under 1 year. So this pollen was collected to be used in controlled pollinations to outcross darling. So this is a really important part of the restoration project in increasing genetic diversity. Because like I said, you don't want to just plant a bunch of clones. You want to capture as much remaining American chestnut genes as you can that are still surviving, because that's how trees protect themselves from all sorts of things. So it's really important to outcross these now at this stage in the restoration process. About 50% of offspring will inherit the oxo defense gene when we do these pollinations based on where the gene is sitting on the chromosome. Here's what it looks like. We take these microscope slides that have all the transgenic. When I say transgenic, it just means the trees that have the oxo gene in them. We take their pollen and we apply it to female flowers out in the field. And that's how the pollinations are performed. You can also see here's a close-up image of the pollen. They're pretty cute. I think they look like little loaves of bread. And we wanted to also do some studies to see how the gene was being inherited and then exposed to the fungus. Are the offspring of this blight-tolerant tree also showing resistance to the fungus? So here you can see to the left a transgenic American chestnut, so one that has the oxo gene that it inherited through doing these pollinations. And after 13 weeks after it was inoculated with the fungus, you can see there the wound is there, and then you can see this orange part right here. So the fungus is there. You can still see it, it's visible, but it's pretty much just reduced to like a surface wound on the tree. And then when we compared this to trees that were also pollinated but did not inherit the oxo gene, so these are just wild type regular American chestnuts, this is what their cankers looked like 13 weeks after being inoculated. So the differences between the response between trees that have the oxo gene and the ones that do not is really dramatic. And that's all just from one gene that was inserted into the genome of the tree to give it this dramatic protection against the fungus. And then here's just a graphical representation of pretty much the same information. Here's two American chestnuts that inherited the oxo gene, so they have significantly smaller cankers compared to trees that did not inherit the oxo gene. And then again, the Chinese chestnut shows kind of this intermediate resistance response too, which is pretty interesting. And then over here to the right again is the pictures of the different kinds of responses in their cankers. So right now we are still working on outcrossing and making these trees more genetically diverse. But the darling chestnuts, the trees that have the oxo, are currently going through federal regulatory review. So because these trees were produced through genetic engineering, they are highly regulated by the government. So we're not allowed to plant them outside of our research areas. We're not allowed to let them open flower. We have to autoclave everything. We can't, make any mistakes in that regard until the government, until they can deregulate us. So if they approve the tree and deem that it is safe, then we can, move on from that and go forward. But the trees have to go through all three of these government agencies, the USDA, the FDA, and the EPA. They have to go through the FDA because, of course, we can eat chestnuts, so it qualifies as being a food product. So recently, we just submitted a massive document to the USDA, and people were able to comment on this if they were in support or if they were against the trees. So it was really helpful in moving this process forward. So we're still waiting to hear back about the USDA, but currently the trees are just at our research stations where they're permitted to grow. And a lot of the information that we had to submit to these government agencies were biosafety analyses to make sure that there weren't any differences that the oxo gene was giving the American chestnut trees. So it wasn't having any adverse or any like really dramatically positive effects on the surrounding environment. We wanted to see that they were no different than the wild type American chestnut trees, but that one oxo gene that was inserted out of the 40,000 or so genes that are in American chestnut, that one wasn't going to have any adverse effects. There were all sorts of biosafety analyses that were done. Transgenic American chestnut leaf litter was fed to tadpoles. Transgenic pollen was fed to honeybees or bumblebees. The leaf litter was decomposed, and the germination of native plants was evaluated. And mycorrhizal studies were done. So this gene was meant to protect against the nasty fungus, but we wanted to make sure that it wasn't going to have adverse effects on beneficial fungi. And pretty much the conclusion from all these safety analyses that were done was that there's no significant difference between wild type American chestnuts and transgenic American chestnuts, which was really good to see. And right now, we're still focusing on outcrossing. So a really cool feature of using this pollen is that you can rescue the surviving genotypes of American chestnut trees. So if you're permitted to work with the transgenic pollen, you can then use this pollen to go to trees that are still surviving and still producing flowers, and pollinate them with them, and or the surviving wild type populations. And then pretty much 50% of all those pollinations, you'll get blight tolerant seed. So all of that seed can then go on, and you can continue to maximize outcrossing. And then, like I mentioned earlier, capture all of the genes and alleles in the surviving population that will make this tree really resilient into the future. Here's just kind of like another way to look at the breeding plan is that we want as little of the darling genome that we started with, this founder genome, to be in the population, because like I said, we don't want clones. We want as much diversity as possible. So every time you cross the tree with a new American chestnut, you're getting rid of more and more of that original genome. And then hopefully what you'll end up with after many generations of outcrosses is that you'll get a really diverse population of American chestnut, but you'll retain that oxogene that will give it that dramatic protection against the fungus. So that's where my job comes in. I manage the controlled pollinations for ESF's research team. So we do these every summer. We try to incorporate as many new mother trees as we can. It's always really exciting at our research station when a new tree is able to produce female flowers, because we can do more pollinations. Chestnuts are considered monoecious, so they have both male and female sexual organs present on one tree individual. But these tree individuals aren't self-compatible. So you can't just have one singular chestnut tree in your yard and expect it to produce fertile nuts. It needs to have some sort of compatible tree near it that's pollinating it. So we have to do all of these controlled pollinations to get fertile nuts. So you can see here, it's one of our pollinations. And if you look right at the tip right here of the style of the female flower, you can see pollen is being used to pollinate these trees. The pollination season every year at ESF is quite a big effort. It takes a lot of teamwork to get these trees pollinated. We often have to use these bucket lifts. Some of the trees are getting pretty tall and we can't reach them with ladders. So you can see part of our research station right here. All of these white bags that are on the tree are female flowers that have been pollinated. So we use the bags for two purposes. One is to, of course, protect the pollinations because they're highly valuable to us. And also because it's part of our regulation process, we can't have any stray pollen being blown around. So everything needs to be really contained and controlled. And here's one of our collaborators working with the trees. This is a little bit just of an up close what the pollination bags look like and everything. And then part of my job is to kind of think about how our pollen collections and pollinations are going to be in the future. So that's why I've been beekeeping part-time for the project, is because we think we might be able to even use things like honeybees to help us with pollen collection, because they're really good pollinators. So they can really, they're like little flying combs in the air. So they're really good at packaging it and processing it. And you can put one of these beehive traps or pollen traps on the front of a beehive, and it'll bring in all sorts of pollen. And you can find American chestnut pollen in there pretty easily. It has like a very distinct golden color. And you can actually collect this pollen and do pollinations with it if you smear it onto like a microscope slide. So that's something that we're looking at in the future as like a really efficient and quick way to collect pollen. And we've also been working with a local drone company, which has been exciting. So we were thinking about working with drones to reach American chestnuts that might be a little bit more isolated or hard to get to. So if there's one that's in the forest and it's flowering, you can't really get to it, that it might be possible to use drones to deliver blight tolerant pollen to it eventually if it's deregulated by the government. So this drone is actually loaded with American chestnut pollen, and we did one of the first studies with it this year, which is pretty exciting. And then after the pollinations are done, we have to bring all the trees, we cut them, or bring all the bags in, we cut them down from the tree using pole pruners. Everything has to be brought into our lab for processing as part of the regulation. And everything's harvested indoors. So this is a really fun time of year because we do it, well, not this year, obviously with COVID, but in previous years, we do like assembly line style, like someone would crack open the bag and the chestnut, and then another person would count the nuts and another person would label them. But it's really exciting because you get to see kind of the outcome of your pollinations and how successful you were. It's really nice to see like something like this where there's 3 fertile chestnuts. That's typically the number of fertile nuts you'd see in one of those spiky chestnut burrs on the tree. But it's sometimes you only get 1 or 2, and sometimes there's none in there, which is a bummer, but it happens. And then all of these nuts are given an individual ID number so that they can be really easily tracked from harvesting all the way from planting into the field. So the trees the nuts from here are stored for a few months in cold storage because they need a dormancy period to be able to germinate. But before we move them outside, we have to test them to see if they inherited the OXO gene for blight resistance. So the way we do that is we can use either leaf or nut tissue from the tree. And it's based on this formula here. So if the tree inherited oxalate oxidase, the gene, in the presence of the nasty fungus here, it'll again break down into hydrogen peroxide and carbon dioxide. So we have a chemical solution that has the oxalic acid in it, and also a chemical that will react with hydrogen peroxide. So if hydrogen peroxide is being produced, it will stain the tissue dark black. So if OXO is there and it's breaking things down into hydrogen peroxide, the tissue will be really obviously black. And it's a really easy way to test to see if that singular gene is there and if your tree will have increased resistance to the fungus. And if it didn't inherit the OXO gene, it will be just regular normal tissue in there. There won't be any staining occurring. So we do this for every single one of our chestnuts that gets pollinated. And then, this is also what the nut tissue staining. So that previous slide was leaf tissue. And then with nut tissue, it's even more obvious. So you can really see how dramatic the staining is on the trees that inherited this OXO gene. So a lot of the nuts, now that they're becoming more genetically diverse, we're planting a lot of these in our research area and in our restoration forest, which I'll talk about in a second. And then some of our most genetically unique are new mom trees. The offspring from them that are most unique go back into that highlight growth treatment that I mentioned to get them to produce pollen. So this is a way that we can really quickly get to the next generation if we can minimize the gap in between reproduction. So the pollen from the new trees will then go on to keep producing more outcrops of trees and keep increasing that genetic diversity. So here is one of our planting days out of the field. It's always a really exciting day. We plant out, so all of these were trees from our previous, so this is from the 2019 pollination season, all of these seedlings. So we usually put in a few different plots at our research station each year. And then this year was an especially exciting year for our research project because last year we received a grant from the Templeton World Charity Foundation. So this grant allowed us to start our first ever restoration forest site. So this allowed us to pretty much transform a section of our research station so that we can study the trees under different kinds of terrains and in an established forest already, because right now all of our trees are growing just pretty much in orchard style setting. So now that we have this grant and we're able to, make changes to the land and increase, our research area, we can study the trees in a more natural setting. So some of the studies that we're working on right now is looking at chestnuts, like if we were to just plant a chestnut right into a mixed hardwood stand, so monitoring their growth in that setting, as well as in pure chestnut stands. So some of the areas were clear cut, which is in this picture. This area was cleared for a 100% pure American chestnut stand, so we can study that type of growth. And then as well as a bunch of other different studies in these restoration demo sites. So monitoring how mycorrhizal fungi, the recruitment of that as they return once the chestnuts are reintroduced, and other types of studies like a co-cultivation study. So that means like growing different kinds of plants or agroforestry products along with the American chestnut like they do in Europe. So there's a study that's currently being set up where it's growing American chestnuts with grapes for winemaking, which is of course really exciting for us. But it'll be important to do these kinds of studies to kind of learn how these trees will, restore and what to look for and stuff, and learn more about them. That's just a few. There'll probably be other kinds of studies with vernal pools and amphibians and reptiles and mammals and birds. So it's a really exciting kind of first step towards restoration. And then of course, we don't want people going out and clear cutting, nice established forests that have, or replacement dominant trees like oaks and stuff. We don't want our chestnuts to be, inserted and cleared and everything. So we envision, future restoration in spots like mine reclamation, so areas that had heavy mining occur, and that this could be a plant that you could grow there to help restore the plant communities, historical sites, private landowners, of course. So really, ESF has produced this tree, but it's really going to be the work of tree lovers and tree enthusiasts who are going to be kind of, at the front of this restoration project, doing these plantings, taking care of them, and doing the first plantings of them until they can reproduce naturally and spread, normally without the assistance of humans. So in the future, we hope to be able to distribute blight-tolerant American chestnuts materials. This is, of course, all pending on the government's decision. So we hope to be able to send people pollen for doing their own outcrosses. So if you have an American chestnut tree growing near you, or you know of one, you can go out and hopefully be able to reach some flowers and start producing blight-tolerant baby trees that way. We also would like to send out scion wood for grafting. So if you know how to graft trees, sometimes you can get them to flower quicker if you graft onto an older rootstock. So that's an option. And then of course, just blight tolerant nuts and trees for direct planting too. So just to kind of conclude and just some, to wrap up some thoughts on everything, darling chestnuts with the oxogene are going to be the first genetically engineered trees to be used for conservation purposes. So I know there's a lot of mixed feelings on how people feel about genetic engineering or GMOs. I think it's always important to kind of review things on like a case by case. And it's really comforting to a lot of people to know that while this is a GMO and genetically engineered, and this hasn't been done before, it's probably one of the most well-studied trees now from all of the biosafety analyses that we've done. So it's just been a really incredible research effort that has gone into making sure these trees are suitable for restoration. And these trees have also been shown to significantly protect American chestnuts from the damaging effects of the blight-causing fungus. So the result of all this is that you're getting a 100% pure American chestnut tree, except for that one gene that has been added for resistance. So it's a completely native species, which is really cool. And then I always like to say that our research project and genetic engineering itself is just a piece of the puzzle. There's not one facet of a research project that's going to be the silver bullet. Conservation problems are really complex, and it takes a bunch of different facets for things to be successful. So things like our research project, along with all the other work that people are doing to protect this beloved tree species, so like the TACF back crossbreeding program, also like more genomic work. So right now there's been a lot of research going into looking for additional genes for blight resistance. The more we understand about the genome of these plants, it can be helpful into the future. So this includes even genes that haven't been studied enough yet within the Castanea genus itself, like some genes from the Chinese chestnut. Like those ones could be useful for durability of this tree into the future. And then, of course, the people that are going out and looking for surviving American chestnut trees is a huge part of it. And the people that keep mother trees alive, that they plant them, and make sure they keep them protected from the blight fungus, which there's people in the audience today like Gail who have been doing the work to keep mom trees alive for when, you know, blight tolerant pollen becomes available. So it's all one big effort from all sorts of different types of research, and it's all super important. And it can also serve as a template for other future plants that are threatened by pathogens and pests. So if this sounds like something that you're super excited about, if you're like really amped to help the American chestnut and you want to get involved, I always recommend people becoming members of the New York chapter of the American Chestnut Foundation. So the foundation's really cool. They have meetings and workshops annually. Of course, we didn't get to have one this year, but we do them virtually. ESF sometimes hosts pollination workshops, so we've done that in the past. So you can come to our campus and learn how to pollinate the trees, so that if you were interested in receiving pollen from us, if the trees become deregulated, you can know exactly how to do it and work with the trees. There's volunteer tree plantings, and of course you can receive seedlings if you're a member of the foundation. So if you wanted to start caring for some wild type American chestnuts that could be pollinated in the future, or if you just wanted to have them now because they're cool trees, that's something you could get from the foundation. And then if you're interested in following ESF updates, you can join our listserv for updates. So you can contact chestnut@esf.edu or myself to be on this listserv, or you can just contact me if you have questions or want to talk chestnuts. I'm totally open to any sort of chestnut discussion. Or you can check out our website or our social media, which I've listed the handles of that. So with that, I just want to say thank you guys so much. You guys have been such a fabulous audience. I'm kind of monitoring my time. I don't think I have a little time to spare. This is good. Now I'll actually turn it over to Gail. I've included a picture of her tree. She's going to talk a little bit about the local American chestnuts in the area.
Yeah, well, thank you, Hannah. That was a great presentation of a really amazing project. I'm really excited to be a part of it. Something I wanted to mention about the slide that Hannah showed early on of the vintage pictures, the historical picture. The picture on the right-hand side that you saw that had the children standing near the base of the tree was actually a tree where we know where that was. It was along Hurley Mountain Road in Hurley, New York. And I remember Dr. Powell mentioning that that's the only photograph of a mature chestnut tree that he knows of in New York State. So it's a pretty important picture, and we were happy to provide it to the project for their presentations.
Awesome.
And okay, and what I wanted to let the group know about is that back in 2015, we planted a small orchard of these mother trees of wild type, open pollinated American chestnuts at Spring Farm. And this is a picture of where they are. They have the $1 million view. And if you wanted to go visit them, we planted 10, there's 8 still surviving. Two didn't make it past the first year, and now some of them are getting the blight already. So we're trying to keep them alive, but we may have to coppice some where you cut it down and let it sprout up again from the roots. So we're hoping that they survive long enough that when Hannah's group can start handing out the blight-resistant pollen, that once I get permission from the preserve, we would be able to pollinate these mother trees at Spring Farm to get, and then half of those resultant nuts would be able to produce a blight-resistant tree. So that was sort of the plan. But even if these trees don't survive that long, it was practice in growing these trees, and it's also people can come visit them. They're located at Spring Farm in a field that's past the barns. If you go to the Spring Farm trailhead, park there, and then hike out to the barns, there's a weather station there in the first field. If you walk past the weather station to the second field through a opening in a stone wall, that's the field where these trees are growing. And they have cages around some of them, so you'll know where they are. So if you want to go visit them when you're on the preserve, you can do that. They'll be blooming in late June or July. They're not getting female flowers yet. We've only seen male flowers, so they're not quite mature yet. Paul, I remember contacting Paul Huth early on before I planted these, and he took us down to the corner of this field that has this wonderful view of the Catskills and showed us a huge log in the corner of the field that was from a chestnut that had grown there. So we know chestnuts grew there in the past. And Dr. Powell visited this site, and he commented, he looked around, he's standing there looking around saying, this is ideal habitat for chestnut. It's got acid soil, it's well drained, it's got a nice slope, there's plenty of sun for the seedlings to grow. And so we were excited to be a part of it. Paul also looked into the historical records of the Mohonk Preserve and found in the Smiley family minutes of 1913, they reported that chestnuts were dying all over the ridge and they didn't know why. So when Hannah showed that picture of the expanding range of the blight, it hit the Shawangunk Ridge in 1913. And we know that because the Smiley family takes these amazing records of their family meetings. So if every year, every spring, I get nuts from Alan Nichols, who's the president of the New York chapter of the Chestnut Foundation. And so I'll be growing some seedlings this spring. So if any of you would like a pair of seedlings, you could contact me. And if I have enough, I'll hand them out to you in, say, May, probably. They'd be large enough to hand out. So you could contact me through my email, and I'll put that on the chat line. It's gswist@gmail.com, but I'll put it in the chat line. And that's really all I wanted to cover. I'd like to turn it back to Hannah to answer any questions that came in on the chat. I see there's a few interesting questions. So, Hannah?
Thank you, Gail. Yeah, at this point, I can take any questions you guys might have. I saw that the chat was kind of blinking at me, so I could either read that, or if you guys just want to ask them right now, either works. But yeah, I'm here. We can discuss.
Yeah, there are a few questions in the chat. There's one from Jeremy. He asked if 50% of offspring inherit the oxo-defense gene, does that mean it's a recessive gene? And so thus, once it's in the wild, will only 50% of the darling offspring be protected?
It's a dominant gene. So any pollen, so when, I'll get a little biology here. When the chromosomes separate during meiosis, if we remember back to like Bio 101, where that separation is occurring when you're creating haploid cells, the oxo is either situated on the way it's situated on a chromosome, and it separates. So some of the pollen, the haploid pollen, will be containing that oxo gene, and then others won't. So it's about 50% based on that separation that have the oxo gene, and then it's dominant. So all the trees that are pollinated that get, when it recombines during fertilization, the offspring that had the oxo gene, when it recombines, will have the blight tolerance gene for defense. And it expresses at different levels, we're finding. So it's not going to be equal expression of the gene in all, but we are still seeing really good results from the majority of the offspring so far that can have resistance to the fungus. Hope I answered that correctly.
Sounds good to me, yes. So another question from Jen is there a hypothesis for why the native Chinese chestnut species was more susceptible to the fungus than the American crossbreed?
Oh, that's such a good question. Yeah, you guys have good ones. I believe it was because there's different levels of pathogen resistance in any natural population of a tree. So that was a specific genotype that we were using. It's known as the king Chinese chestnut. So it could be that one's just not as resistant to the fungus as some of the other Chinese chestnuts. So if you were to test like the Nanking genotype, which is said to be the most resistant Chinese chestnut genotype that they've documented, you might see higher levels of resistance there. So I think it's just because there exists like kind of like a spectrum of resistance to the pest. So that one, seemed to have had intermediate resistance.
And that's really it for the questions. There's just a comment about from Betty. She says, I may be mistaken, but there are a lot of chestnuts spread on a number of sites on the preserve and Minnewaska. She gave an example of one on Millbrook Ridge Trail. It keeps sprouting but dies back. And from the sounds of things, it probably there are quite a few of these tiny chestnuts, the stump sprouters. Am I correct, Gail? Do you have that info?
There are a lot of American chestnuts, stump sprouts all over the preserve. Actually, Paul Huth was studying those with Dan Smiley way back when, back in the 70s, I think 1970s. And they actually did a study of 10 areas they mapped out to where there were stump sprouts. And they've been tracking those. I think they rechecked them around year 2000. And then I think again more recently. And what they're finding is that those stump sprouts are smaller and fewer as time goes by. So even though the tree is still alive, technically it's not really a part of the ecosystem like it was before, and it's dying out. So if we don't do something like what Hannah's group is doing, we won't have a chestnut at all. I mean, we're lucky that we still have enough of the diversity still alive in the forest. And we just hope that we have enough of those that are growing to fruiting age, that or flowering age, that we can capture their genotypes.
Mm-hmm.
Yeah, so there you'll see small shrubby sized chestnut trees here and there in the preserve. Sure.
Yeah, I admittedly, my tree idea is not spectacular. So a lot of times I'll overlook American chestnut trees that are naturally still around. Mm-hmm. So if they still flower, for me, it's easiest to go on hikes in October, because it's really easy to see the burrs on the ground. So that's like a good ID for finding a chestnut species, is looking for those burrs if they're able to flower still. Obviously, it's tougher if they're shrimpier and not mature anymore, but that's a good little trick.
There's a couple of things just came in.
I know, I'm like trying to read and like listen at the same time.
Well, Karen was wondering if we could see the photo of the American chestnut in New York again that Gail referenced.
Sure.
Yeah.
Share my screen again.
Sure, yeah.
Oh, yeah, it was early on.
This guy?
It was one of your first pictures. Yeah, this next.
Oh.
Chart 3, I think. Chart 3, I think he was asking about. The historical picture. Back at the beginning. Yeah. Chart 3. The one before that, 2 before that.
Oh.
Yeah, no, this one on. That picture on the right is a tree. Well, okay.
There you go.
Yeah, that tree was growing on Hurley Mountain Road. Those barns behind there were on the Henry Paul Farm, which is now called Row by Row Farm. So if you drive, the site is still there. The tree is long gone, of course. And actually, the Hurley Mountain Road was slightly diverted in the 20th century. So Hurley Mountain Road is actually on the far side of the tree, going on to the right of the barns and to the left of this house that you see to the right. So we know who those people are. It's a really remarkable picture, and it was taken by a photographer from Schenectady, who was related to one of the DeWitt descendants. The DeWitt family lived there at the time, the Dutch family, the DeWitts. And he was president of the Schenectady Camera Club at the time. And so he's somebody who thought to take a portrait of a tree, which people didn't think of doing that back then. This was done in 1890s, around 1890. And so it's a really remarkable picture. Really, really happy that we have it. Definitely.
It's one of my favorites for sure.
And we believe, now show the picture of the painting, it was 2 down, I think. We believe that tree, that Hurley tree, was the model for the Winslow Homer engraving that you see on the left. Chestnutting, it's called, because it's a bifurcated tree, just like the Hurley tree was. And that slope to the right behind the tree, we believe is Hurley Mountain. And then the fields would be off to the left. So we're pretty sure that based on the timing when that engraving was done, we know Homer was in the Hurley area at that time in 1872. So or 1870, I think it was. So, so that tree was a model for that engraving by Winslow Homer. And the watercolor that you see in the middle is also by Winslow Homer. It was done later in 1878, but could have been in the Hurley area or somewhere in the Adirondacks. We're not sure where that tree was. It was also a Homer work.
So cool. Yeah, it says by Winslow Homer on the chestnutting one. You guys probably can't see. It's because the text is covering it. But
yeah,
so cool.
Yeah, yeah. Well, we have just a few more moments here. So let's see. There is a question from Matt. He says, do you think similar application could be done for other trees being affected by other diseases like the ash tree or the elm tree?
Yeah, great question. Actually, we want to extend this type of research to other species because we have all the tools to do this kind of work now. So we have a postdoc actually that's working on the American elm tree. The elm tree is threatened by two different species, two different pathogens. So one is the Dutch elm disease, and that one can actually be selective breeding can be used to help that tree. But then there's a second disease that affects the elm called Dutch elm yellows, I think. And then that one's trickier because it's I think it's a fungus. It's either a bacteria or a fungus. So our postdoc is actually looking at some genes that could be inserted into the elm to give it resistance to the elm yellows disease. So that's a new one that we're branching off on, possibly ash too. We haven't started any research on the ash, but we are working with the chinkapin species. So earlier in the presentation, I mentioned how there were two other North American species that are in the same genus that were affected. So those are two of the ones that our research group is also looking at because it's so similarly related to the American chestnut. It should, in theory, be easier to move on to those trees next as another tree to work with. So, yeah, good question.
Yeah, yeah. And then we'll do one final question from Jeremy. Is there an idea when the FDA might respond? And are there precedents for similar genetically modified tree approvals that kind of give a sense of the timeline?
No, and that's what's kind of tough because we're pretty much the first non-agricultural group to go through these regulatory processes. So they've never seen anything like this before because it's the first tree ever for conservation purposes, at least in our country, that have tried to go through this process. So I'm not sure about the FDA. The USDA, they always give an estimate of between 1 and 3 years before they'll have a final decision for us. We haven't submitted anything yet to the other two federal agencies, in part because I think COVID is slowing everything down. So it's just been tougher to work with them, but that's the next aim is the EPA and then finally the FDA, but those ones haven't been officially submitted yet.
Nice, nice. All right, well, it looks like that's probably the end of our questions. So thank you, Hannah and Gail. This has been a really great presentation. It's kind of fun to kind of go down memory lane a little bit with my tree studies there. So yeah, and those photographs are amazing of those chestnuts. It really does bring it home about how large those trees were and how important they were to our forests, and now they're gone. So thank you both for presenting and for the great work that you've been doing, and thank you everybody for joining us this evening for our webinar series that we're doing here. There will be more in the future. If you are interested, I mean, this is a talk on trees. So next month there is a forest bathing talk webinar that we'll be doing. So if you want to get out and do some forest bathing and maybe find those chestnuts at Spring Farm, that's the webinar for you. Yeah, so I want to thank you all again, both for coming here and for presenting this evening.
Thank you so much. Thanks for having me. This was really fun. Appreciate it.
Thank you so much. That was great.
Thank you.